friends, welcome to Veena's Educate. In the previous video, we learnt about nutrition, types of nutrition, and the most important thing, the universal food providers, yes, the plants, which provide food for all the organisms on this earth through a process known as photosynthesis. Imagine if we could also undergo this process known as photosynthesis, then we wouldn't have to struggle for food all this life. But we are not gifted with this process. Only the plants are the organisms on this planet which are gifted by this process known as photosynthesis. It is told, Paropkaraya phalanti vrikshaha. That means trees, plants, they prepare food to serve the planet, to serve the organisms on the planet. So this is Veena, a teacher by profession. I am going to give you content videos on the subject biological science, English and motivational videos. So let's get started. Photosynthesis, a very vital process which takes place only in plants. But there were many scientists. They kept on pondering over years to find out actually how this process took place. What are the materials the plant required to prepare food for all the organisms? All these things the scientists kept on pondering. And then, since after 300 years, there came a scientist known as, his name was von Helmont. He wanted to know actually how the mass of the plant increases. What is responsible for the increase in mass of the plant? So what he did is, he just took a potted plant. In that he planted a willow tree. And then he noted down the mass of the pot, the soil in the pot, and then the mass of the willow tree. The mass of the soil was 90.72 and the willow tree mass was 2.27. Then what he did was, he covered the soil with a sheet which had holes. And through these holes only air and water could enter. And then he left this tree for 5 years. He did not do anything. He did not touch. And he just left the tree as it is for 5 years. And after 5 years, he measured the mass of the tree. He found that the willow tree, the mass of the willow tree increased to 76.74 kgs. And the soil mass was 90.66. So when we minus the final and the initial readings, we find the mass increased by 74.47 kgs and the mass of the soil 0.06. The willow tree, it grew only on rainwater. So with this he concluded that plant water is very essential for the growth of plant mass. Then came another scientist. His name was Joseph Priestley. He wanted to know what is the relationship between air and the plants. So what he did is, he just took a bell jar. He took a bell jar and then he introduced a candle into the bell jar. A burning candle into the bell jar. And then he also introduced a rat. He introduced a rat and then he left, he just wanted to see what would happen. He found that after some time, the candle in the bell jar got extinguished and the rat also was no more alive. Okay, it was no more alive. Then what he did is, he just introduced, in the next part what he did, he just introduced a mint plant. Okay, he introduced a mint plant and then he again lit the candle. He again lit the candle and he put a rat. He found that for many hours together, the rat was alive in the bell jar and even the candle also was burning very brightly in the bell jar. So he understood that there must be some relationship between the plant and the animals, the things to burn. He learned that the gas which was released by the candle and the rat, it was not good for for themselves, but it is good for the plants. And what the gas was released, the gas which is released by the plants was useful by the candle and the rats. So he learned that the plants and the animals, organisms and the things which are required to burn all are interdependent. So that means he understood that plants release some gas which is very, very important for the organisms to live and for the candle to burn. So both of them, they exchange the gases, and the carbon dioxide which is released from the candle and the carbon dioxide which is released from the rat is used by the plant, and plant releases oxygen. This oxygen is used again by the candle to burn and for the rat to live alive. So this is how Joseph Priestley. Now we shall again learn. Then came another scientist, C.B. Van Neel. He was C.B. Van Neel. He conducted experiment on purple sulfur bacteria. Purple sulfur and then he learned through when he worked on purple sulfur bacteria, he learned that light is very, very necessary for photosynthesis. Then what he did is, instead of H2O, he used H2S, hydrogen sulfide. He learned that during photosynthesis, element sulfur was released. Then later on came another scientist known as Robert Hill. What he did is, he showed that oxygen is released not from H2S but from H2O. Then these scientists, they formulated a simple equation for photosynthesis which goes this way. 6 molecules of carbon dioxide plus 12 molecules of water in the presence of sunlight and chlorophyll. It gives one molecule of glucose, C6H12O6 plus 6 molecules of H2O plus 6 molecules of oxygen. So this was the final equation of photosynthesis, and this equation is very, very important. During the exams, this question can be asked for 1 mark or for 2 mark. Write the equation of photosynthesis. When you are asked to write the equation of photosynthesis, you will write it as this way. Now, from this we also learn that these are the raw materials for photosynthesis. What are the raw materials? Carbon dioxide, water, sunlight and chlorophyll. These become the raw materials for photosynthesis, and the end products of photosynthesis is glucose, water and oxygen. This shows that as a result of photosynthesis, glucose is formed, prepared, and then water is released, and even oxygen also is released into the atmosphere. So important this photosynthesis process is. And this glucose which is prepared by the plants, later on it gets transformed to starch or to cellulose, and then plants also prepare some other forms like synthesize certain other forms like proteins and fats. Now what we have to learn is, scientists have already proved that this is the equation for photosynthesis, but how do we believe it? We need to prove that really during photosynthesis glucose is prepared, oxygen is released or not. So for this we shall do a small experiment and then find out whether photosynthesis is during photosynthesis glucose is prepared and oxygen is released or not. To find out the presence of starch in leaves. Now let us learn whether during photosynthesis glucose is prepared or not. For that we will do a small experiment. The aim of the experiment is to test the presence of starch in leaves. For this, what do we require? We require an apparatus of this size. So we have learnt in photosynthesis that glucose is prepared, produced during photosynthesis, and but we need to prove it whether glucose is prepared or not. For that we need to do a small experiment, and this experiment shows presence of starch in leaves. The aim of the experiment is to show the presence of starch in leaves. For this we require an apparatus like this. Now I'll tell you what are these. The materials. This is a Bunsen burner. This is a Bunsen burner. And this is a tripod stand. And this is the test tube. This is the asbestos gas. This inside the test tube we have methylated spirit. This is the beaker. This is water in the beaker. And then we have a leaf in the test tube. And then now we have here, we have a petri dish. Petri dish, and this is iodine, or you can also say betadine solution. Iodine, or you can take betadine solution. So these are the materials required for this experiment. Now what do? Is we take a beaker full of water, and then in the beaker we place a test tube. The test tube will have, we place methylated spirit in the test tube, and a leaf is inserted into the test tube, and then this beaker is burnt over this Bunsen burner. It is heated over the Bunsen burner, and till when it is heated? It is heated till the leaf turns into pale white in color. Now once the leaf has turned into pale white color, then the leaf is removed carefully by the brush and spread over the into a petri dish, and iodine solution is added to the leaf. When you add iodine solution to the leaf turns into blue-black in color. This shows that you know that any carbohydrate, when it is tested with iodine, turns into blue-black color. So when the leaf is turned into blue-black color, this shows that the carbohydrate, that is glucose, is prepared by the leaves. So with this experiment we prove that leaves produce starch, or leaf produce glucose. Now here you can be asked with two questions. The one question is, why should we boil the leaf to pale white in color? You need to boil the leaf until it turns to pale white in color to remove the chlorophyll from the leaf. Another question which could be asked is, why can't we heat the test tube directly on the flame instead of placing the test tube into a beaker? So as you all know, this test tube is filled with methylated spirit, and when we heat the methylated spirit directly on the Bunsen burner, it may catch fire. So hence this test tube is placed in a beaker full of water to avoid this catching of fire. So with this experiment we prove that leaves prepare starch. Starch is present in the leaves. So now we will go to the next experiment. Now let us learn whether during photosynthesis we had learned that oxygen is also released. Whether it is released or not, we have to prove it through an experiment. So today we shall prove it through an experiment. That is, oxygen is produced during photosynthesis. The aim of the experiment is to prove that oxygen is produced during photosynthesis. For that, the materials required is, we require a beaker. This is a beaker, and then inside the beaker we have water. Then we have a funnel here. This is a funnel, and inside the funnel we have hydrolate twigs. Here we have a test tube. Then we also require incense stick or a burning splinter. Now what do? Is we have to arrange the apparatus in this way. Take few hydrolate twigs and then place it in a funnel, and place this funnel in a beaker full of water. Now, and ensure that the water in the beaker should be above the stem of the funnel. Now take a test tube, fill it with water, and then invert the test tube onto the funnel. And then leave this, arrange the apparatus in this way, and leave this apparatus in the sunlight for nearly 3 to 4 hours. After 3 to 4 hours, you will start observing small bubbles start arising from the funnel, and they go upwards and they start getting collected at the end of the test tube. Once a sufficient amount of gas is collected in the test tube, carefully what do we have to do is, we have to hold the test tube with the help of our thumb, remove it, and then bring a incense stick. Incense stick means you can also say agarbatti or burning splinter. Bring that agarbatti or burning splinter in front of the test tube. You will find that this incense stick or the burning splinter, it burns with very brightly. It burns very brightly. This shows that the gas which was present in the test tube is nothing but oxygen, because oxygen is the only gas which supports things to burn, and when there is oxygen, things burn more brightly. So when the burning splinter or the incense agarbatti was brought in front of the test tube, it started burning more brightly. So this experiment proves that during photosynthesis, in the presence of sunlight, oxygen is also released by the plants. So with this we have learnt that how photosynthesis, the equation for photosynthesis. We have also learned that during photosynthesis glucose is prepared. We have also learned that oxygen is also released, and all this we have not simply learned, we have learnt it through experiments. So now in the next video we shall learn about the different factors which affect photosynthesis or which are responsible for photosynthesis.